Hello and welcome to our online audience, and thank you for joining us today for this Science AAA's webinar, Deciphering Immuno-oncology, Targeting Innate Immunity in Cancer. My name is Sean Sanders and I'm Senior Editor for Custom Publishing at Science. Today we present the second installment in our four-part Deciphering series. You can access the first part by clicking the link just above the slide window, or by going to webinar.sciencemag.org. Also, look out for parts 3 and 4 in this series coming up in September and November this year. In today's webinar, we're going to be looking at the role of innate immunity in cancer. Although targeting T cells has proven to be a technically successful treatment for most cancer immunotherapies, only a fraction of patients respond. There has recently been a surge of interest in investigating the relatively underexplored innate immune system as a possible tool for therapeutic intervention. Innate immune effector cells, including natural killer cells, macrophages, and dendritic cells, have been shown to interact with cancers and inhibit their progression. Dissecting the molecular details of these interactions will aid in identifying cancer-derived intrinsic factors that can be exploited to develop effective immunotherapy regimens. Our speakers will provide examples of how innate immunity pathways are involved in fighting cancer and how these pathways might be co-opted to generate new treatments. I'd like to welcome those speakers now. They are Dr. Santiago Zelenay from the Cancer Research UK Manchester Institute in Manchester, UK, and Dr. Stefani Spranger from the Massachusetts Institute of Technology in Cambridge, Massachusetts. Thank you both so much for joining us today. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a View Bio link, which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we'll have a short Q&A session, during which we will address some of the questions submitted by our live online viewers. So if you're joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box and clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology for sponsoring today's webinar. Now, I'd like to introduce our first speaker, Dr. Santiago Zelenay. Dr. Zelenay joined the CRUK Manchester Institute in 2015 as a junior group leader to form the Cancer Inflammation and Immunity Group. His work focuses on understanding the underlying mechanisms that mediate cancer inhibitory versus tumor-promoting inflammation, with the goal of designing new therapies for cancer patients. Welcome, Dr. Zelenay, and thank you so much for being with us today.
Thank you very much, Sean, for the invitation to participate in this webinar and for the opportunity to present our work here. So indeed, as Sean just introduced, the revolution we are experiencing in cancer treatment these days is truly justified by the remarkable and unprecedented responses some patients experience following therapies aimed at harnessing the anti-tumor activity of the immune system. Strong and sometimes durable responses have been observed in patients with advanced metastatic disease, and not just in highly immunogenic cancers such as cutaneous melanoma, but also in cancer types considered until very recently as refractory to immune-based therapies. However, invariably, only a fraction of patients benefit from treatment, while many show no benefit, and only a very limited proportion of them experience complete responses. The underlying basis that accounts for this lack of response in some patients is arguably the most important open question in the field, highlighting the need for mechanistic studies to understand this lack of responses. So my talk today will focus on a proof-of-concept story that essentially started with a very basic and fundamental question, which was, what makes cancer or malignant cells immunogenic? What are the features that control specific immunity against them? Indeed, to understand how cancer cells evade immunity, we first need to understand how they trigger or initiate immunity in the first place. But the question of how tumors evade immune responses is indeed a very long-standing question in cancer biology. And this slide here illustrates some of the properties of malignant tumors that were shown to enable them to outgrowth by avoiding recognition and elimination by both innate and adaptive arms of the immune system. It is, for instance, well known that some cancer cells, and this is here in the upper left, can avoid to be taken up by phagocytes by a regulation of the, for example, so-called do not eat me signals. Equally, we know that some tumor cells are weak stimulators or actually actively inhibit innate immune cell activity. And here I'm depicting a dendritic cell where we know that in some cases tumor cells very poorly stimulate these cells, or they actively suppress their activation or the migration into the tumor draining lymph node. A very well-known process of immune evasion, well described, is that one here in the upper right, in which the tumor cells actually lack or lose the expression of strong relevant target rejection antigens. So even if they are presented by antigen-presenting cells, if there's not a cognate strong antigen to be seen by the T cells, that immune response will be inefficient. And finally, even when all these processes do take place, we now know that in some cases, at least tumor-specific responses do take place, but the effector response is actively inhibited at the tumor site by exclusion of cytotoxic effector cells, or by a regulation of inhibitor receptors such as PD-L1 in tumor and non-tumor infiltrating cells, or by the presence of a highly immunosuppressive tumor microenvironment. I would like to introduce briefly the role of inflammation in cancer. So the concept that cancer can induce inflammation and that inflammation can induce cancer is very well established. And indeed, inflammatory cells at the tumor site can promote several key aspects of malignant tumor growth, such as proliferation, invasion, or angiogenesis. Indeed, subtypes of macrophages, T cells, and neutrophils and other cells are commonly found infiltrating tumor cells. And these cells produce mediators, chemokines, growth factors that can directly or indirectly support cancer growth and progression. This is what we typically call as cancer-promoting inflammation. On the other hand, there's also another type of flavor of inflammation is that one in which the tumor cells and the type of inflammatory cells that we have in those tumors are typically associated with anti-tumor immunity and the production of factors that are typically associated with good prognosis and response to treatment. So that's what we call cancer inhibitor inflammation in this context. But going back to our original question, we were intrigued, or what drive our question was, what is actually making these cancer cells immunogenic? What are the properties that allow them to initiate an immune response? And we originally decided to study a cancer cell line that was isolated by a genetically engineered mouse model developed by Richard Murray and colleagues, in which induced expression of the most common mutation found in cutaneous melanoma in melanocytes in these animals leads to the development of melanoma in these mouse models. And we focus our study on studying the properties of cancer cell lines isolated from those animals that are immunocompetent. So our hypothesis or assumption that was cancer cells isolated from these mice must have evaded natural tumor immunity in the original host that is immunocompetent, and therefore are likely to retain key attributes that allow them to avoid immune-mediated recognition. So in Bob Schreiber, we thought these cells are probably immunoedited. So to start to study the immunogenic properties of these cells, what we did is simply take these cell lines and implant it into immunocompetent C57 black 6 mice that are gene-genic with the original mouse model. And what you can see here in this plot is that when we implanted those tumor cells into wild-type animals here in the black circles, these tumors invariably form progressive tumors. And when we did the same experiment in RAG-deficient animals, so mice lacking adaptive immunity, these tumors also grow progressively, indeed showing that adaptive immunity has a very little impact on the growth of these tumor cells. And as I mentioned before, this is somehow consistent with the idea that tumor cells might be poorly immunogenic because they lack a strong target antigen to be recognized. Indeed, the loss of tumor-rejected antigens is one of the best studied immune escape mechanisms. However, a central paradigm in immunity is that in order to induce an effector cytotoxic T cell response, those naive T cells, and in this case are depicting CD8 positive T cells, not only need to see a cognate antigen presented in the surface of antigen-presenting cells, but also those antigen-presenting cells need to be activated. And by this, I mean not only they need to express co-stimulatory molecules such as CD8 T or CD86, but they also need to produce cytokines that promote the differentiation and effector function of those cytotoxic T cells. And we know a lot about what are the signals and pathways that drive this activation in the context of bacteria, viruses, and fungal infections, but much less is known in the context of cancer or malignant cells. So our hypothesis was that perhaps these melanoma cells do contain some strong tumor rejection antigen, but indeed they might be suppressing the activation of innate immune cells such as dendritic cells. So to test that possibility, what we did is basically take bone marrow derived cultures from mice, where basically we took these dendritic cells, different flavors of them, or primary macrophages in culture, and we incubated them in the presence of conditional medium from these Braf-driven melanoma cells grow in standard condition. And we did these cultures in presence or absence of lipopolysaccharide to induce the activation of those myeloid cells. And the first striking phenomenon that we found when doing these cultures is that by simply adding conditional medium from these melanoma cells into these dendritic cell cultures, what we found is that these cells started to produce substantial amounts of IL-6, CXCL1, and GCSF, to name but a few. And these are cytokines, growth factors, and chemokines that are typically associated with what I defined before as cancer-promoting inflammation and are associated with bad prognosis and lack of response in treatment. On the other hand, when we did these experiments in presence of lipopolysaccharide, as expected, this induced the production of TNF, IL-12, or MIB-1 alpha by these dendritic cells or myeloid cultures. But now when we did this stimulation in presence of conditional medium from these melanoma cells, now what we saw is a profound inhibition of the production of these factors. So we were very, very intrigued to what could be the factor that is in this conditional medium that is promoting this strong modulation of dendritic cell and macrophage activity. And because this conditional medium was actually, so this modulatory activity was resistant to heat inactivation, protease treatment, and DNase and RNase treatment, we started suspecting that this could be a lipid. So this is we came with a lipid-mediated hypothesis, and indeed, many of the results that I just described were reminiscent of previous studies showing that indeed, in some cases, cancer cells can produce prostaglandin E2, an inflammatory lipid mediator, and that can have strong modulatory activity on diverse immune cells. So prostaglandin E2 belongs to a family of prostanoids that is produced downstream of the metabolism of arachidonic acid and is produced by the rate-limiting enzymes cyclooxygenase 1 or cyclooxygenase 2, COX-1 and COX-2. And indeed, cyclooxygenase 2 is known to be overexpressed in several types of human cancers, including prostate, colorectal cancer, breast cancer, and many others. And both cyclooxygenase 2 and prostaglandin E2 in itself have been directly implicated in several aspects of malignant tumor growth, such as proliferation, invasion, migration, and angiogenesis. So the first thing we did is actually check whether these melanoma cells were actually producing prostaglandin E2, and this is what you can see here on the left. This is an ELISA for prostaglandin E2, and you see that when we put wild-type cells, so the conditional medium from the wild-type cells, there's very high levels of prostaglandin E2 in this conditional medium. Now, so then to test and to actually prove that this was actually mediating the modulation of the activity of dendritic cells and macrophages in our cultures, what we did is to CRISPR out cyclooxygenase 2, or both cyclooxygenase 1 and cyclooxygenase 2 in these cancer cells, and this is what you can see here in the Western blots on the right. So indeed, as expected, cells that are deficient in cyclooxygenase 2, or both cyclooxygenase 1 and cyclooxygenase 2, are impaired in their ability to produce prostaglandin E2, and consistent with our hypothesis that was responsible for the modulation of dendritic cell activity in these in vitro cultures. If we now take conditional medium from the wild-type cells, as I showed you before, this induces production of IL-6, so this is in the central panel. You can see that when we take conditional medium from the Braf-driven melanoma cells, cyclooxygenase is competent. We get a lot of IL-6 being produced. I showed you that before, but now if you use conditional medium from the cyclooxygenase deficient cells, these cells now, this conditional medium is now unable to induce production of IL-6, and is also unable to inhibit the production of TNF that is induced by lipopolysaccharide, here on the right part of the slide. But of course, our question was, what happens with the immunogenic properties of these cells in vivo and the growth profile of these tumors? So what you see here, and as I showed you before, if you take, these are the black circles, when we take these cyclocygenase-competent cells and we implant them into immunocompetent animals, these cells grow progressively and form large tumors. But now when we take the cyclocygenase-deficient cells and we put in immunocompetent animals, these cells form tumors, but then they spontaneously get rejected. And this was perhaps not so surprising, as I just mentioned, cyclocygenase has been involved in inducing cancer cell proliferation, promoting angiogenesis, and inhibition of apoptosis. However, the striking finding was that when we put the cyclocygenase-deficient cells into mice that are immunodeficient, again, the RAC-deficient mice that like both B cells and T cells, these tumors form progressive tumors, really demonstrating that cancer cell intrinsic activity, the cyclocygenase intrinsic activity by the cancer cells is essential and dominantly for immune evasion, and these cells can really form normally vascularized tumors regardless of whether they cannot produce the prostaglandins. Moreover, we were interested to understand what happens in the tumor side in terms of the inflammatory profile of those tumors preceding the effect of the T cells in these tumors. So for that, what we did is to look at these tumors formed by the cyclocygenase-competent and the cyclocygenase-deficient very early on, 4 days upon implantation into the mice. And what you can see in this slide is that what we found is that when we look at tumors formed by cyclocygenase-competent cells, they had an immune signature that is associated with, I defined before, as cancer-promoting inflammation, characterized by production of factors such as IL-6, CXCL1, I1 beta, and others that were induced by prostaglandin E2 in our in vitro cultures. In contrast, tumors formed by the cyclocygenase-deficient cells not only had an immune signature associated with cancer inhibitory inflammation, so not only had a reduction in IL-6, I1 beta, or CXCL1, but now had a concomitant increase in many factors that are typically associated with anti-tumor immunity and effector cytotoxic T cell function, including things like the granzymes or chemokines that are typically associated with infiltration by CTLs into the tumors. So this leads. So the next thing we did is to see whether it was a peculiarity of this melanoma cell line that we were studying. So we extended these studies to other cell lines. So the first here, what you see is the BRAF melanoma cell line in the upper left, is the one I just showed you the results. We also extended those studies to another melanoma cell line that we obtained from Richard Murray and colleagues, in which these cells are driven by oncogenic NRAS. And exactly as what we saw for the BRAF-driven melanoma, what you can see in the upper right here is that those cancer cells, when they are wild type, so they express cyclocygenase, they form progressive tumors, but when they are immunocompetent animals, and we ablate the expression of cyclocygenase, the cyclocygenase-2, and the gene that encodes for cyclocygenase is PTGS2, so PTGS2 deficient cell lines are actually grow as the parental cells, but then in immunocompetent animals, they invariably get rejected, while if they are implanted in immunodeficient animals that lack adaptive immunity, they grow progressively as the parental cell lines. Exactly the same, although in this case we didn't obtain full rejections, but a strong reduction in the proliferation and retardation in the proliferation we obtained when using commercially available colorectal cancer CD6 cancer cells and the breast cancer 41 cells, typically known for very poorly immunogenic, and in both cases by ablating the ability of the cancer cells themselves to produce prostanoids by ablating cyclocygenase-2 in these cells. What we see is that those tumor cells, when implanted in immunocompetent animals, they grow much slower, but then when you put in mice that are deficient in T cells, so in nude mice, these cells form progressive tumors exactly as the parental cell lines, showing really that in these 4 tumor cell lines, cyclocygenase activity by the cancer cells is primarily essential for evading immune recognition and elimination. So this leads us again to the model in which we think that when the cancer cells themselves express cyclocygenase, they produce this very strong immunosuppressive factor, which is prostaglandin E2. What it does is fuels tumor-promoting inflammation, and those tumors are typically characterized by high levels of IL-6, CXCL1, and GSF, and leads to something that it looks like a very poorly immunogenic progressive tumor. But when we ablate cyclocygenase expression on those cells, these cells are unable to produce prostaglandin E2, and now those tumors are characterized by a tumor inhibitory microenvironment with high levels of IL-12, CXCL10, and interferon gamma, and they are also getting infiltrated by completely different types of cells that are known to be associated with responses to immunotherapy. And for mechanistically, we have extended these studies and in terms of what is going on in these tumors, and I invite you to look at a recent study by Jan Bocher and Quechano Reyes de Sousa, where we describe what really we think is going on within these tumors. and when there's actually immune-mediated tumor control. Now I wanted to move into something that shows how this proof of concept study had actually led to something that we think has very clearly potential therapeutic implications, which is this idea that by manipulating the type of inflammation we have within tumors, we can actually raise the cancer immunogenicity and potentially enhance the response to treatment. So this figure depicts here that we have the two types of inflammation that I described before, and our working model is by using cytotoxicity inhibitors such as NSAID and other anti-inflammatory drugs, we can perhaps switch this inflammatory microenvironment into the tumor, making it more permissive for responses and for attacks by T cells. So this is what we originally did in this study, and it's basically going back to the melanoma model. What we did here is to treat tumors that were bearing the BRAF mutant melanoma lines with anti-PD-1 blockade, with aspirin, a very well-known COX-1 and COX-2 inhibitor, or with a combination of both. And what you can see in these plots is that when we use aspirin alone, there's basically no effect in the growth of these tumors. When we use anti-PD-1, there's a moderate significant effect in this particular cell line, but when we combine both treatments, both aspirin and anti-PD-1, we get an enhancement in the mouse, in the number of animals that experience full rejection of the tumors, and also an acceleration in how fast that response happens. So another way of looking at the same results is if we look in these same experiments 3 weeks following treatment, and this is what is shown in this slide. What you see is that when the mice are untreated, all tumors are quite decent in size and homogeneous. So what you see here in the untreated in the black tumor. So each dot is one animal. At this time point, when the mice are treated with anti-PD-1, we see no significant effect at this point. Only some mice start to show some sort of response. Aspirin by itself doesn't do anything. But then when we combine both aspirin and anti-PD-1, at this time point, 40% of the mice already fully rejected those tumors. And this response by the combination of both is really dependent on the immune system, because if we do this treatment in mice, again, that lack adaptive immunity, the treatment fails completely. So more recently, we have extended these studies to other anti-inflammatory drugs, and in this case, I'm showing the results for celecoxib, a known and a very well-known COX-2 specific inhibitor, and in this case, this is with the C26 colorectal cancer model, where you basically see again, when these mice are treated with the vehicle, these tumors form progressive tumors. Invariably, when we treat them with celecoxib, and these are the red curves, there's absolutely no treatment and no effect in terms of tumor growth control. When we use anti-PD-1 monotherapy, we do see consistently a few mice responding to this treatment, but most of them do not respond. But now when we combine anti-PD-1 and celecoxib, we find a remarkable synergy with most of the mice fully rejecting these tumors. And here on the right, you can see a compilation and quantification of many of these experiments, where we see that only with the combination, we get a very significant effect with almost 75% of the mice experiencing full and complete responses. So this is all preclinical models, but of course we wanted to see to what extent this could be validated into human, in case of in humans. So we are extending these studies to many tumor types, where we found there is a conservation in terms of how this cyclosation pathway controls the inflammatory status and the immune profile of those tumors. But here what I'm depicting is our initial analysis of a cohort of cutaneous melanoma patients, and what we did in this case is basically stratify those samples with those having high levels of cyclosation S2 and those with low levels of cyclosation S2. So what you see in this picture, each column is a patient. So what you can appreciate is that in the upper left, those with high levels of cyclosation S2, they also have high levels of these tumor-promoting factors that were induced in the mouse model by prostaglandin E2 and were associated with cyclosation-competent cells. And while in contrast, those that have low levels of cyclosation S2 not only have reduced levels of these tumor-promoting factors, including CXL1, I1 beta, or GSSF, but now what we see also is an increase in many anti-tumor mediators, which expression is actually a good sign when you find it in tumors. So these include many interferon-stimulated genes, and also things like CD8A or CD8B that indicate that these tumors are potentially more infiltrated by T cells. So as I said, we have extended this to other tumor types. This seems to be the case. But with this, I would like basically to mention some of the conclusions and implications of this proof of concept study. And the first one is that I hope I convince you that cancer cell-derived prostaglandin 2 can enable progressive tumor growth by modulating the inflammatory profile at the tumor site. Another important implication of the results I just show you is that relevant target antigens can be retained within cancer cells thanks to tumor-derived immunomodulatory factors. I think this is quite an important implication because it indicates that when the cancer cells themselves produce immunosuppressive factors, they are potentially able to carry more genetic aberrations that in absence of those immunosuppressive factors can be recognized by the immune system. Finally, we think that the expression of a specific inflammatory signatures in biopsies can be associated with cancer patient outcome and response to treatment. And finally, this idea that inhibiting the cyclooxygenase 2 or prostaglandin 2 pathway can act synergistically with anti-PD-1 blockade, and this provides a rationale to test this combination in the clinic, and us and others are currently testing that possibility. And with that, I would like to finish by acknowledging all members of my group at the Cancer Research UK Manchester Institute, Richard Marais for strong collaboration and providing some of the mouse models that we use in this study, and a special thanks goes to Cristiano Reis Sousa at the Francis King Institute and members of his lab where I initiated this work, many other collaborators and past and current collaborations of my group, and Cancer Research UK and other funding bodies for funding the work in my group. And with that, I would like to thank you for your attention.
Thank you so much, Dr. Zelenay. We're going to move along to our second speaker now. That is Dr. Stefani Spranger. Dr. Spranger is at the Koch Institute for Integrative Cancer Research at the Massachusetts Institute of Technology. Her work seeks to elucidate the mechanisms and pathways underlying the interaction between the immune system and cancer, using mouse models that recapitulate the co-evolution of tumor progression and the anti-tumor immune response. The research aims to shed light on new or improved immunotherapies for human cancers. Welcome, Dr. Spranger, and thank you very much for being on the line.
Sean, thank you very much for this kind introduction, and thanks for the opportunity to present our work here today in this webinar. And I'm going to tag along with the very nice presentation from Santiago and trying to convince the audience that Batf-3 dendritic cells are key to induce a potent anti-tumor T-cell response. And Batf-3 dendritic cells are also commonly called DC-1s or danger dendritic cells or COX-9A positive dendritic cells. So the rationale for us studying dendritic cells in the first place came from an observation that patients do have sometimes ongoing anti-tumor immune responses characterized by CD8 T cells within the tumor microenvironment. And this you can see here on the very left upper plot in patient 1. Staining for CD8 in a section of a malignant melanoma patient. The presence of those CD8 T cells often coincides with the recruitment and upregulation of immune inhibitory cell types as well as pathways. And this is here indicated by staining for FOXP3 suggesting the presence of regulatory T cells, as well as PD-L1 and IDO, two very strong immune suppressive pathways upregulated in the tumor microenvironment. In stark contrast, patients also occur that do not have this ongoing anti-tumor immune response present in the tumor microenvironment. And those patients often lack the immune inhibitory pathways that I just described. Using a preclinical mouse model, we were subsequently able to show that the presence of CD8 T cells, and in particular the interferon gamma produced by those CD8 T cells, is essential for the upregulation of those immune suppressive mechanisms. And this is shown here for IDO, which is failed to be upregulated, as well as failed upregulation of PD-L1 if either CD8 T cells are depleted or we use an interferon gamma deficient host. This was subsequently also shown to be a key observation in patients for response against checkpoint blockade therapy. In this case shown here, the group around Tony Ribas and Paul Tomei at UCLA have segregated patients treated with anti-PD-1 immunotherapy and segregated based on response or progression upon therapeutic intervention. And when they then subsequently looked in pre-treatment biopsies, they found that the responders had a higher degree of CD8 T cell infiltration within the tumor microenvironment compared to patients that progressed. And all of those preclinical as well as clinical studies have by now shed light on the T cell inflamed tumor, and we now have a fairly good understanding on how the T cell inflamed tumor is implemented and why T cell inflamed tumors actually progress. And this is due to upregulation of immune inhibitory pathways. And those immune inhibitory pathways are actually the pathways we're currently targeting with immunotherapy, and that makes sense that only those patients are responding to current checkpoint blockade therapies. In contrast, as Santiago already nicely highlighted, we have very little understanding of how a non-T cell inflamed tumor microenvironment actually is being induced and can be maintained. And our approach to investigate this was to look for tumor cell intrinsic signaling pathways that potentially directly impact the local tumor microenvironment. And this notion arose from the fact that we actually in the clinic had observations that patients do have T cell inflamed as well as non-T cell inflamed lesions present within the same patient, suggesting that this is a tumor intrinsic rather than a host mediated mechanism. And to investigate this, we used the TCGA dataset for malignant melanoma, and we used a gene signature containing 13 different genes that we knew was predictive for the presence of tumor-reactive CD8 positive T cells within the microenvironment. And using this gene signature, we were successful in segregating the malignant melanoma patients into the ones that have a T cell signature low profile and the ones that have a T cell signature high profile. And we only focused on the very low expressing group 1 through 3 or the very high expressing group 6 through 8. And then subsequently used those two cohorts to perform pathway analysis using the Ingenuity platform, as well as mutational analysis looking for gain and loss of function mutations, and combined this with downstream target gene analysis. And all of these analyses led us to one signaling pathway that was differentially activated, and that was the activation of Wnt beta-catenin within the non-T cell inflamed patient cohort in melanoma. And we then went on and wanted to generate a mouse model which would allow us to conduct mechanistic studies, whether or not tumor cell intrinsic activation of Wnt beta-catenin could actually mediate lack of T cell infiltration. And to do that, we used a genetically engineered mouse model, which is based on a Cre-ER cassette under the tyrosinase promoter. And this limits the expression of Cre-ER to only melanocytes. This was combined with a mouse model with Cre-dependent activation of BRAF V600E, as Santiago pointed out, the most common driver mutation in human melanoma, and was further combined with a floxed version of P10, accelerating the PI3 kinase pathway. And this was further combined with a stabilized version of beta-catenin, and to give us a tool model antigen, we also combined this with a SIY reporter so that we would have a tumor-specific antigen that we could use for immunological mechanistic studies. And of course, our initial question that we wanted to address is, would this mouse model recapitulate what we found in the human patients? And indeed, we saw on the very left here, looking at BRAF P10 mice, we did saw that those tumors had an anti-tumor immune response infiltrating into the tumor, indicated here by staining against CD3 positive T cells. In contrast, if we combine BRAF and beta-catenin or use the triple BRAF P10 and beta-catenin, we lost this CD3 positive T cell infiltrate into the tumor microenvironment. And we were also able to quantify this using flow cytometry shown on the bottom. Of course, our notion was that this mouse model would recapitulate sensitivity towards checkpoint blockade therapy. And to test this, we used the same mice that I just described, induced tumor, waited 4 weeks, and then exposed those mice to combination therapy using anti-CTLA-4 and anti-PD-L1 blocking antibodies, given every other day for the duration of the experiment. And as you can see on the left, in the BRAF-P10 setting, so the tumors that do have a T-cell infiltrate in the tumor microenvironment, those tumors actually respond quite nicely to checkpoint blockade therapy. And in contrast, if the tumors were T-cell deprived via the beta-catenin pathway, those tumors did not respond to checkpoint blockade therapy anymore. And this nicely highlighted to us that we actually now have a model in hand which recapitulates the responder versus non-responder phenotype observed in human patients. We then wanted to know what is actually different about those two tumor models, particularly focusing on the immune side of things. And we observed, looking at the antigen-presenting cell compartment, that the beta-catenin positive tumors lack CD8 alpha positive and CD103 positive dendritic cells from the tumor microenvironment. And if you want to look to the very right of the slide, you can see the flow plot, and on the left flow plot, you see this double positive or single positive population of dendritic cells present, and this population is entirely ablated in beta-catenin positive tumors. And those dendritic cells are commonly driven by the transcription factor Batf-3 and hence will be referred to as Batf-3 dendritic cells. Those dendritic cells have been known and have been described previously to be extremely important for cross-presentation and uptake of tumor-derived antigen and migration towards the tumor-draining lymph node. So our next test was, of course, whether or not those tumors would have an impaired T-cell priming when beta-catenin is activated. And to investigate this, we used, again, the same mouse model and then adoptively transferred naive T-cells labeled with a proliferation dye. And those T-cells were specific for the model antigen, which we have engineered into our mouse model SIY. And this allowed us to first track activation of T-cells in the tumor-draining lymph node, as well as recruitment of T-cells into the tumor microenvironment. And when we looked within the tumor here on the left side of the slide, you see if SIY is present, those SIY-specific T-cells are actually accumulating in the tumor microenvironment, but only in the context of the T-cell inflamed tumor, and not in the context of the beta-catenin positive non-T-cell inflamed tumor. And this is accompanied and reassures that those Batf-3-driven dendritic cells are key for T-cell activation if we looked in the tumor-draining lymph node. In the tumor-draining lymph node, if SIY, the antigen, is present, T-cells are accumulating, and as you can see on the very right part of the slide, you can see that CFSE is actually being diluted, indicating T-cells are proliferating and have been activated in an antigen-specific manner. And in contrast, on the bottom right part, you see the T-cells are not accumulating in the tumor-draining lymph node, and CFSE is not being diluted, indicating that transfer T-cells are not seeing the cognate antigen. And this led us to just summarize this part, this led us to the observation that those Batf-3-driven CD103 positive dendritic cells are needed even in those genetically engineered mouse models to uptake tumor-derived antigen and bring it to the tumor-draining lymph node. And this is how an early anti-tumor immune response could be blunted. But over the course of the last couple of years, many more studies have now indicated that other tumor cell intrinsic signaling pathways besides beta-catenin can also ablate dendritic cells from the tumor microenvironment or alter their functions. And one very nice example that Santiago just gave is tumor cell intrinsic activation of COX-1 and 2 signaling pathways. So we now wanted to probe the possibility whether or not we could overcome this lack of T-cell priming by just giving adoptive T-cell transfer into those mice and bypass the need for the expansion in the tumor-draining lymph node. And when we did this experiment, on the left part of the slide, you see here again the T-cell inflamed tumor. We again observed a very highly therapeutic benefit in the T-cell inflamed cohort, the Braf-P10 mice. But when we did the very same experiment of adaptive T-cell transfer in the beta-catenin positive non-T-cell inflamed tumor mice, we did not observe any therapeutic benefit in those animals. And since we were puzzled by this experiment, we then wanted to fully reassure that we're observing a real phenomena and established a new imaging approach where we could implant a skin flap window on top of those autochthonous melanoma model tumors and visualize tumor lesions in the presence or absence of adaptively transferred T cells. When we did that, on the left part of the slide, you see a T-cell inflamed beta-catenin negative tumor. The tumor cells are here depicted in green, and the T cells are depicted in red. And as you can see, the T cells are crawling in very close contact across the tumor and engaging with many tumor cells over the course of this movie. In contrast to this, in the non-T-cell inflamed beta-catenin positive tumor setting, only very few T cells can be observed, and you can see here only 2. And those T cells seem to be either stuck in the tumor matrix or even in the extracellular matrix and cannot interact with the tumor cells directly. This observation was further reassured when we came back a couple of days later, and here you see on the left a tumor lesion 48 hours after adaptive T-cell transfer. And again, you see the green tumor cells and the red T-cells interacting in very close contact, and this is in the T-cell inflamed setting, of course. And then when we came back 3 days later, the tumor was entirely eradicated, but the T cells are still patrolling through the former to be tumor lesion. And this very nicely highlights the potency of adaptive T cell transfer and the potency of immunotherapy, because it's most likely the only therapy that will continue to be active despite the fact that the therapy itself has been discontinued. We further also wanted to really boost the system to the very far end and did a vaccination approach prior to tumor induction. And when we vaccinated those mice against the SIY antigen and subsequently induced SIY negative tumors, this vaccination, of course, had no effect on the tumor growth. But if we did the vaccination approach in the setting of a T-cell inflamed tumor, we saw a very, very stark reduction in tumor outgrowth, indicating that there is an adaptive immune response induced in this setting. And again, even a strong vaccination approach was not capable of driving effector T cells into the tumor microenvironment of beta-catenin positive tumors. And this then led us to ask the question on why those effector T cells are not migrating into the tumor microenvironment, and what key elements need to be present in a T cell inflamed tumor to drive effector T cell migration. And to do that, we went back to our original model, the Braf-P10 and the Braf-P10 beta-catenin tumor mice, and sorted out tumor cells, stroma cells, as well as the entire antigen-presenting cell compartment. And we focused in on CXCL9 and CXCL10 because we knew from previous work that those are the key chemokines required for effector T-cell homing into the tumor microenvironment. And this, although the tumor cells are making some level of CXCL9 and CXCL10, the predominant source of those chemokines could be found in the antigen-presenting cell compartment. And since we knew that the beta-catenin positive tumors are lacking Batf-3-driven CD103 positive dendritic cells, we then compared conventional dendritic cells with CD103 positive dendritic cells, and indeed we found that the predominant source for CXCL9 and CXCL10 are those cross-presenting Batf-3-driven dendritic cells. To further prove the requirement of those dendritic cells for effector T-cell migration into the tumor, we did an adaptive cell transfer approach, transferring bone marrow-derived dendritic cells that resemble those Batf-3-driven dendritic cells into the tumor microenvironment. And when we combined this with adaptive T-cell transfer of effector T cells specific for the SIY antigen in the non-T cell inflamed tumor, this was the only condition where we could actually induce successful effector T cell migration and homing towards the tumor. But we also wanted to prove necessity of those dendritic cells, and to do that we had to do a quite elaborate approach where we used the BRAF P10 SIY mice as host mice and generate mixed bone marrow chimeras. Those mixed bone marrow chimeras were a mix of either wild type BLAK6 bone marrow plus CD11 CDTR bone marrow, and this allowed us to, after tumor development and after initiation of an anti-tumor immune response, deplete the CD11 CDTR fraction of the dendritic cells and being remained in the upper case with wild type dendritic cells. But in the lower case, we mixed the CD11 CDTR bone marrow with bone marrow from Batf-3 knockout mice, and those mice lack CD103 positive dendritic cells. So upon depletion of the DTR positive dendritic cells, we're left with a dendritic cell compartment that lacks Batf-3 dendritic cells. And the following experimental scheme was done after the bone marrow was engrafted. We induced the tumor, and 21 days later we depleted the DTR-expressing dendritic cells by giving diphtheria toxin. And subsequently we gave effector T cells specific for the SIY antigen, and then just evaluated recruitment into the tumor microenvironment. And here you can just see that we were successful in depleting those CD103 positive Batf-3 driven dendritic cells in the experimental setting, and this depletion of dendritic cells entirely ablated the recruitment of effector T cells into the tumor microenvironment, indicating that those dendritic cells actually have a dual role. For one, as I showed you in the first part of my talk, they're key to prime CD8 T cells in the tumor-draining lymph node. But we now also identified that they're key for recruitment of effector T cells back into the tumor microenvironment. And this makes tumors that lack those Batf-3-driven dendritic cells a very hostile and hard to treat tumor type for immunotherapy. And the next investigations will likely go into identifying therapeutic avenues where we can enrich for those dendritic cells and increase the frequency of those dendritic cells in tumors in order to make those tumors more susceptible towards immunotherapy. And with that, I would like to thank my current group here at MIT, as well as my postdoctoral mentor, Tom Gajewski. And I'm handing it back to Sean.
Wonderful. Thank you so much, Dr. Spranger. Thank you to both of the speakers for their really fascinating presentations. We're now going to move along to questions submitted by our online viewers. So a quick reminder to those watching live, you can still submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. So the first couple of questions, I'm going to come to you, Dr. Zelenay, with a few specific questions about some of your work. The first is, this viewer asks what the status of the dendritic cell was in co-culture. Were they naive or differentiated? And I'm not sure if you'd like to maybe talk about the reason this question might be asked.
Oh, yes. Well, I guess I suspect, so in this case, we use different flavors of dendritic cells. So we have used FLT3 ligand-derived dendritic cells, which contain some of those equivalents of the C103 that now Stefani nicely described. But we've also done the same experiment with GM-CSF-derived myeloid differentiated populations, including monocytes and dendritic cells, and more recently also with primary MCSF-derived macrophages. So in all those cases, they were naive at the time of the stimulation, but we also added LPS in certain cases to stimulate their activation. So essentially, they were naive in terms of they were not actively activated, simply differentiated with growth factors as is typically done. But we did promote their activation. We also showed that this is also the case within the tumors. So if we look inside the tumors in the mice, in the presence of tumor formed by cycloocygenase competent cells, those CD103 DCs that Stefani nicely described just now were also not activated in terms of the expression of costimulatory molecules, but in the absence of cycloocygenase, they increase their presence in the tumors, and they are also activated, and they produce things like IL-12.
Great, thank you. The second question I have for you is, what is the concentration of COX-2 used in your tumor models, and how will you address the safety concerns using anti-COX inhibitors and anti-PD-1 treatment? Since this viewer says anti-COX-2 inhibitor seems to have a lot of liabilities in rheumatoid arthritis treatment.
Indeed, and indeed this is one of the issues with this treatment. So, for example, we don't think the working model is that we really need a reduction in the levels of prostaglandin 2 intratumorally, so we would have to go for high doses of aspirin to test this, or for really high doses of selective inhibitors. So we would have to potentially use inhibitors that are tackling the pathway downstream without affecting the production of other prostanoids, or use more selective inhibitors that are more safely tolerated. And those studies are on the way. But indeed, we think our working model is that we really need to reduce the levels of prostaglandin 2 intratumorally, and for that we probably need high doses of inflammatory mediators.
Dr. Spranger, I'm going to come to you with a couple of questions. The first one is, how important is CXCL9? Since this viewer says most groups seem to focus on CXCL10 as the CD8 positive T cell recruitment in tumors.
There's certainly a large degree of redundancy because the receptor for both of those chemokines is CXCR3. In our hands, we've seen, depending on the mouse model that we're actually looking, sometimes CXCL10 is higher expressed and sometimes CXCL9 is higher expressed. From biological sequence, it is very likely that CXCL9 is very early in tumor development and might be more profound in transplantable tumor settings, because a lot of those experiments are done day 3, 4, 5 after tumor induction, versus CXCL10 seems to be a little bit later in the immune response, and that's why it might be picked up more in genetically engineered mouse models and even human patients.
Thank you. The next question I had for you is, this viewer says that they thought that T cells required a certain sustained physical interaction in the minute range with target cells to function effectively. And they ask, what is the real time, the real amount of time of contact of the T cells infiltrating the tumor cells?
So in our hands, we hardly have found T cells that are not in contact with tumor cells at any given time. When you look really closely at those movies, you can actually see that the T cell is still in contact on the bottom part of the T cell with one tumor cell, and then starts to engage with the next tumor cell while crawling along. So I think we'll need more in vivo intravital imaging data to actually show this, but my gut feeling would be that T cells are in constant contact with tumor-expressing, antigen-expressing tumor cells.
Great, thank you. Dr. Zelenay, back to you. Considering that small molecule COX inhibitors are so widely used these days, has anyone observed, as far as you know, any fortuitous effects on tumor progression in vivo from such drugs that might be consistent with the results that you found? And I'm guessing they're talking about in humans.
Yes, indeed. After this, something that we're looking for and trying to look in the cohort of patients, those that were actually taking NSAIDs of aspirin. And as I said, our working model is, what we think is that for this to be efficient, you really need to go for high doses, and those are not the ones that people usually take. However, there has been an abstract in ASCO where basically they look into retrospectively of a cohort of patients that were treated with PD-1 blockade, and indeed, despite the cohort was not large in number, that those patients that actually reported that were taking some sort of NSAID, even at low doses, did have a trend to show better responses. So I think this needs to be studied, because again, many of these drugs are over the counter, so you don't need it's actually how to control, and we do need to do a clinical trial for this. But there is already some evidence reported by others that indeed those patients that were taking low doses of some sort of NSAID show a trend to get more benefit from PD-1 blockade, for instance.
Thank you. And just a quick additional question for you, Dr. Zelenay. One of the viewers asked what the tumor model was that you were using for the last set of the COX-2 inhibition studies that you presented.
So we have extended this to many other tumors, but the first set of data was with the melanoma mouse model, the Braf-driven melanoma cells, and the second one where I showed the data with the celecoxib was with the commercially available CD6 colorectal cancer cells. But again, we have tested, and others as well, this combination works in many other tumor types as well.
Okay. Dr. Spranger, back to you. Do you think that, I guess this is asking you to look into your crystal ball or hypothesize a bit, but do you think that T-cell-based companies should be engineering BATF-3 dendritic cells to give alongside T-cell therapies?
We have given our dendritic cells intratumorally. And I think if you want to continue the avenue of providing a cell-based therapy, that would have to be considered as well, which is not a big problem if you have cutaneous or near-surface tumor lesions. If you have deep tumor lesions, that might actually be a therapeutic hurdle to be overcome by surgeons or physicians that are actually delivering the therapy. There are other avenues that people have addressed, and for instance, Miriam Murad's group from Mount Sinai in New York has a very nice, elegant study giving FLT3 ligand systemically and combining it with a local poly-IC administration into the tumor, and this also increased the presence of those cross-presenting Batf-3 positive dendritic cells. So I think we need to be a little smarter about how we could actually target those dendritic cells and how we can activate them and provoke them to overcome their either lack of activation, lack of differentiation phenotype.
And I have another quick question for you, Dr. Spranger. This viewer says they apologize if they missed this, but they're asking what is the molecular mechanism linking the beta-catenin activation with the lack of CD103 positive dendritic cells in the tumor microenvironment.
Yeah, I glanced over this. So molecularly, we know that beta-catenin in the tumor cells leads to the transcription of ATF3. And ATF3 is a transcriptional repressor for many immune modulatory molecules, including CCL4. That's another chemokine. And the expression of CCL4 in our hands, and I think Santiago showed in his work that CCL5, which is a redundant chemokine, does that in the COX-2 positive tumors, mediates the recruitment of CD103 positive dendritic cells.
Okay, I'm going to come back to Dr. Zelenay. We've just got a minute or two left, and perhaps actually both of you would like to comment on this. But Dr. Zelenay, in the inflamed tumor is a predictor of checkpoint response. So this viewer asks, why is PD-L1 not a good prognostic factor?
Well, I guess it's referring to the T-cell inflame, and I guess it's that what we know, and I think this is beautiful work also from Tom Gajewski and Stefani, that, for example, PD-L1 expression comes after T-cell activation downstream of interferon gamma and also by oncogenic signaling. So in some cases, even if you don't have PD-L1 expression to start with, when you activate those T-cells with immunotherapy, those T-cells might produce interferon gamma, and that leads to the upregulation of PD-L1. So even if it wasn't there in the first place, it might still be upregulated, and those patients might still respond to treatment. So presence or not presence is, we still have a lot to understand about what regulates these molecules in there, but as I said, the upregulation of this might happen after treatment, and that can dampen the response as well, even when it wasn't there in the first place.
Yeah.
Dr. Spranger, did you have any thoughts?
Yeah, I 100% agree. And in addition to that, some tumors upregulate PD-L1 independent of the presence of CD8 T cells. For instance, deletion of P10 in some cancer types, for instance, lung cancer, can induce spontaneous upregulation of PD-L1 with no CD8 T cells present, and in this scenario, anti-PD-1 therapy would not be predicted to work because you don't have the T cells that are PD-1 positive sitting in the tumor microenvironment.
That's fantastic, and unfortunately that does bring us to the end of our webinar today, so it just remains for me to thank our speakers today, Dr. Santiago Zelenay from the Cancer Research UK Manchester Institute and Dr. Stefani Spranger from MIT. Please do go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. In particular, please don't miss our next installment in this Deciphering series to be broadcast in September this year. This particular webinar will be made available to view again as an on-demand presentation within about 48 hours from now. We'd be very interested to know what you thought of today's webinar. Please send us an email at the address that's now up in your slide viewer, webinar@SSS.org. Again, thank you so much to our fantastic panel and to Cell Signaling Technology for their kind sponsorship of today's educational seminar. Goodbye.